Today, we'll be discussing on the second part of biomembrane structure and composition, fluidity and asymmetry. Let's have a look at the introduction. In the previous talk, we have discussed that the boundaries of cells are formed by biological membranes, the barrier that define the inside and outside of a cell. This barrier prevents molecules generated inside the cells from leaking out and unwanted molecules from diffusing it. Yet, they also contain transport systems that allow the cell to take up specific molecules and remove unwanted ones. Such transport system confers on membrane the important property of selective permeability. Here, we will be focusing more on the asymmetry and fluidity of membrane. Biomembrane forms an asymmetric structure, and as it is fluid-like, it has its fluidity, that is, translocases with various molecules of the cell. The asymmetry of the biomembrane can be partially attributed to the irregular distribution of proteins within the membranes. This asymmetric organization is important for cell functions such as cell signaling. The asymmetry of the biological membrane reflects the different functions of the two leaflets of the membrane. As seen in the fluid membrane model of the phospholipids bilayer, the outer and the inner leaflet of the membrane are asymmetric in their composition. Membrane fluidity refers to the viscosity of the lipid bilayer of a cell membrane or a synthetic lipid membrane. Lipid packaging can influence the fluidity of the membrane. Membrane fluidity needs to be maintained for the cell to function normally. Example, when red blood cells try to squeeze through blood capillaries. Also, fluidity allows small molecules to diffuse rapidly through and add self-movement, growth, division, etc. The aqueous environment inside and outside of a cell prevents membrane lipid from escaping from the bilayer, but nothing stops these molecules from moving about and changing with one another within the plane of the bilayer. The membrane, therefore, behaves as a two-dimensional fluid, which is crucial for cell membrane function. This property is distinct from flexibility, which is the ability of the membrane to bend. Around 1970, researchers first recognized the individual lipid molecules are able to diffuse freely within lipid bilayer. Two types of preparation have been very useful on such studies. First is the bilayer mat in the form of spherical vesicles called liposome, and the second one is the planar bilayer called black membrane formed across a hole in a partition between two aqueous compartments. This simple artificial bilayer allows delicate measurement of the movement of the lipid molecules, revealing that some types of movement are rare while others are frequent and rapid. The fluidity of cell membrane has to be precisely regulated. Certain membrane transport processes and enzyme activities ceases when the bilayer viscosity is experimentally increased beyond a threshold level. The fluidity of a lipid bilayer depends on both its composition and its temperature. A synthetic bilayer made from a single type of phospholipid changes from lipid state to a two-dimensional rigid crystalline or gel state at a characteristic freezing point. This change of state is called a phase transition, and the temperature at which it occurs is lower if the hydrocarbon chains are short or have double bonds. Bacteria, yeast, and other organisms whose temperature fluctuates with that of their environment adjust the fatty acid composition of their membrane lipids to maintain a relatively constant fluidity. Cholesterols modulates the property of lipid bilayer. When mixed with phospholipids, it enhances the permeability barrier properties of the lipid bilayer. It inserts into the lipid bilayer with its hydroxyl group close to the polar head groups of the phospholipids so that its rigid, plate-like steroid rings interact with and partially immobilize those regions of the hydrocarbon chains closest to the polar head group. At high concentration, found in most eukaryotic plasma membrane, cholesterols also prevent the hydrocarbon chains from coming together and crystallizing. Despite their fluidity, lipid bilayers can form domains of different compositions. Because a lipid bilayer is a two-dimensional fluid, we might expect most type of lipid molecules in it to be randomly distributed in their own monolayer. Next, we'll see the factors determining membrane fluidity. Membrane fluidity can be affected by a number of factors. One way to increase membrane fluidity is to heat up the membrane. The melting temperature, that is Tm, of a membrane is the temperature at which the phase transitions occur. Below the Tm, the lipids are highly organized. Above the Tm, membranes are more mobile and less tightly packed, and the average thickness of the membrane decreases. The membrane phospholipids incorporate fatty acids of varying length and saturation. Lipids with shorter chains are less stiff and less viscous because they are more susceptible to change in the kinetic energy due to their smaller molecular size, and they have less surface area to undergo stabilizing van der Waals interaction with neighboring hydrophobic chains. Lipid chains with double bonds are more fluid than lipids that are saturated with hydrogen and thus have only single bonds. Cholesterol acts as a bidirectional regulator of membrane fluidity because at high temperature it stabilizes the membrane and raises its melting point, whereas at low temperature it interacts between the phospholipids and prevents them from clustering together and stiffening. We'll see the asymmetric structure of membrane. Membranes are asymmetric structure. The lipid bilayer of a membrane are asymmetrical structure. The reason of the cell membrane is asymmetric is because when proteins are synthesized by the pre-existing membranes, they are inserted into the membrane in an asymmetric manner. The asymmetry of the cell membrane allows the membrane to be rigid and allow the cells to have a different intracellular environment from the existing environment. Additionally, the cell membrane are distributed asymmetrically across the lipid bilayer in a phenomenon called cell membrane phospholipid asymmetry. An inside-outside asymmetry is provided by external location of the carbohydrates attached to the membrane protein. In addition, specific enzymes are located exclusively on the outside or inside of membrane, as in the mitochondrial and plasma membranes. There are regional asymmetry in membrane, such as those occur in the villous borders of mucosal cells, are almost microscopically visible. Others, such as those at gap junction, tight junction, and synapses, occupy much smaller region of the membrane and generate correspondingly smaller local asymmetries. There is also inside-outside, that is, transverse asymmetry of the phospholipid. The choline-containing phospholipids, that is, phosphatidylcholine and sphingomyelin, are located mentally in the outer molecular layer. The aminophospholipids, the phosphatidylserine and phosphatidylethanolamine, are preferentially located in the inner leaflet. Obviously, if this asymmetry is to exist at all, there must be limited transverse mobility of the membrane phospholipids. In fact, phospholipids in synthetic bilayer exhibit an extraordinarily slow rate of flip-flop. The half-life of the asymmetry can be measured in several weeks. However, when certain membrane proteins such as the erythrocyte proteins, glycoprotein are inserted artificially into synthetic bilayer, the frequency of phospholipid flip-flop may increase as much as 100-folds. The mechanism involved in the establishment of lipid asymmetry is not well understood. The enzymes involved in the synthesis of phospholipids are located on the cytoplasmic site of microsomal membrane vesicles. Translocases, that is, flipases, exist that transfer certain phospholipids, example, phosphatidylcholine, from the inner to the outer leaflets. Specific proteins that preferentially binds individual phospholipids also appear to be present in the two leaflets, contributing to the asymmetric distribution of these lipid molecules. In addition, phospholipid exchange protein recognize specific phospholipids and transfer them from one membrane, example, the endoplasmic reticulum, to the other, example, mitochondrial and peroxisomal. The asymmetry of the lipid bilayer is functionally important. The lipid composition of the two halves of the lipid bilayer in the membranes are strikingly different. In the human red blood cell membrane, for example, almost all the lipid molecules that have choline in their head groups, that is, phosphatidylcholine and sphingomyelin, are in the outer half of the lipid bilayer, whereas almost all of the phospholipid molecules that contain a terminal primary amino groups, that is, phosphatidylethanolamine and phosphatidylserine, are in the inner half. Because of the negativity charge, phosphatidylserine is located in the inner monolayer. There is a significant differences in the charges between the two halves of the bilayer. Lipid asymmetry is functionally important, especially in converting extracellular signals into intracellular ones. Many cytosolic proteins bind to specific lipid head groups found in the cytosolic monolayer of the lipid bilayer. The enzyme protein kinase C, for example, is activated in response to various extracellular signals. It binds to the cytosolic phase of the plasma membrane, where phosphatidylserine is concentrated, and requires this negativity charged phospholipids in order to act. Phospholipids in the plasma membrane are used in another way to convert extracellular signals into intracellular ones. The plasma membrane contains various phospholipases that are activated by extracellular signal to cleave specific phospholipid molecules, generating fragments of these molecules that act as a short-lived intracellular mediator. Similarly, specific inositol phospholipids are concentrated in the cytoplasmic half of the plasma membrane of the eukaryotic cell. These minor phospholipids are cleaved into two fragments by specific enzymes that are activated by extracellular signals. Animals exploit the phospholipid asymmetry of the plasma membrane to distinguish between live and dead cells. When animal cells undergo apoptosis, phosphatidylserine, which is normally confined to the cytosolic monolayer of the plasma membrane lipid bilayer, rapidly translocates to the extracellular monolayer. The phosphatidylserine exposed on the cell surface signals neighboring cells such as macrophages to phagocytose the dead cells and digest it. From the present study, it can be concluded that the structure of a biomembrane is selectively permeable with a lipid bilayer. It has different composition on one side of the membrane than on the other side of the membrane. The lipid bilayer are asymmetrical in structure, and this asymmetry is important for cell functions. Besides, membrane is also a fluid structure as it has its fluidity. The fluidity is because phospholipids and proteins can move in the plane of the bilayer. The fluidity of a membrane depends on various factors like temperature, cholesterol content, and fatty chains of the polar head groups. All these components affect the fluidity of a membrane, which is very important for the cell to function. Lastly, it can also be concluded that the fluidity and asymmetric nature of all biomembrane is important for various functions of the cell in a living system.